Hi, my name is Schultz, and I am presenting the gut microbiome project today. The microbiome is the types and proportions of bacteria present in a particular environment. We are particularly interested in the gut microbiome. We study the gut microbiome because it carries out important functions in the human body, such as digestion, and has been linked to diseases such as Crohn's, colon cancer, irritable bowel syndrome, autism, and depression. A study was conducted in 2007 by neuroscientist Paul Patterson to study the correlation between prolonged fever during pregnancy and offspring with autism. The results of a study conducted on rats showed that there is a correlation between the two. The mice with the maternal immune activation had abnormal gut microbiomes. So it is concluded that autistic behavior may be rooted in the gut. A microbiologist from Belgium, Professor Rees, conducted a study where he compared the gut microbiomes of humans with and without depression. He found that two kinds of microbes were missing from the depressed group. So in conclusion, he found that there is a difference in between the gut microbiomes of depressed people. So the question that we were experimenting with for our project was, will there be a difference in gut microbiomes between a snake and a bearded dragon? And our hypothesis for our question was, we believe that a difference would occur in the gut microbiome between the species. So the two species that we compared for this project was a 12-year-old corn snake and a bearded dragon. The diet of the corn snake was primarily carnivorous, while as the lizard had a more vegetarian diet. Though the two species have differences, they do share some similarities. They are both reptiles, they both have scales, they both lay eggs to reproduce, they both shed skin, and they're both ectothermic. So the process of analyzing the gut microbiome of the corn snake and bearded dragon includes three major steps. Number one, isolating the DNA. Number two, amplifying the DNA. And number three, analyzing the nucleotides using a sequencer. So first we use the QIAGEN kit to isolate the DNA from the fecal sample. The QIAGEN kit is manufactured by QIAGEN Incorporated, and its purpose, as I said, is to isolate the DNA from the fecal sample. We followed the protocol instructed by the QIAGEN kit in using it. Next, we use the Zymo kit manufactured by Zymo Incorporated to increase the DNA concentration by tenfold. The DNA isolation was conducted following the Zymo Incorporated protocol. We used the NanoDrop 2000 to determine the concentration or purity of our DNA sample. DNA absorbs UV light at 260 nanometers and protein at 280 nanometers. We used the 260 to 80 ratio to determine the purity of our sample. And samples that have a ratio of 1.5 to 2.2 are considered pure samples, and then below 1.5 are likely to be contaminated with protein. Next, we use the polymerase chain reaction, or the PCR, to amplify the 16S RNA gene. We are amplifying the 16S RNA gene because it contains two highly variable regions, V3 and V4, that are useful in determining and classifying the bacteria present in the gut. So the process of the PCR consists of 25 cycles of three basic steps, which are denaturing the protein at 95 degrees Celsius for 30 seconds, and then anneal primers at 55 degrees Celsius for 30 seconds, and then DNA synthesis at 72 degrees Celsius for 30 seconds. So next we conducted the gel electrophoresis, and the purpose of the gel electrophoresis was to determine if our PCR was successful or not. And then we conducted the PCR cleanup. We used the Ampere XP kit manufactured by Agincourt Incorporated, and the purpose for the PCR cleanup is to remove molecules that could contaminate the sample prior to the indexing PCR. Next, we conducted the indexing PCR. The purpose of the indexing PCR is to add a unique set of sequencing tags to the amplicon so that we may identify and analyze the samples during sequencing later. And how the indexing PCR works. During the PCR reaction, the 3' end will attach to the amplicon, while the 5' end will attach to the sequencing tag. And the indexing PCR consists of 3 steps. First we will run the PCR at 95 degrees Celsius for 3 minutes, and next we will do 8 cycles at 95 degrees Celsius for 30 seconds, then 55 degrees Celsius for 30 seconds, and then 72 degrees Celsius for 30 seconds, and then lastly we will run it at 70 degrees Celsius for 5 minutes and store at 4 degrees Celsius. And next we did the second PCR cleanup, which again removes molecules that contaminate. The molecules that we removed were the unused primers, the excess components from the buffer, like the enzymes, and leftover DNTPs. Next, for the quantification of the PCR products, we use the Quibit fluorometer, and it works by the fluorescent dyes attached to the DNA molecules and traces specific shifts in fluorescence for determination. The fluorometric assay is used instead of the spectrophotometer because it's more precise and it can distinguish between DNA contaminants, and it also can read lower concentrations of DNA and RNA. And the last step was the DNA sequencing. We used the MiSeq automated DNA sequencer manufactured by the Illumina Corporation. We used it to sequence the various amplicons in the fecal samples to determine the bacteria present in the gut microbiome of the 2 species. So this is our data from the UV absorbance, and it shows that 3 out of our 4 samples were within the 1.5 to 2.2 purity range. Sample 429, our first trial, was below 1.5, which means that it was likely contaminated with the protein. However, our other 3 readings were pure enough to continue with. So this is the results from our gel electrophoresis, and we were not successful at extracting the DNA from the feces. There was only 1 lane in the 907 in our electrophoresis results, and we expected to see a band in all of our lanes besides the negative control, and we only saw the 1 band in the 907 lane. So these reads are not for the 2 subjects that we performed the steps for analyzing the microbiomes on. Instead, we used samples of 2 dogs from a given database. And so we compared 2 different dogs, subject 333 and subject 520. Subject 333 had 14,406 successful and total reads, and then subject 520 had 165,179 successful and total reads. And these numbers were large enough for us to adequately base our analysis on. And as for the positive control reads for our 2 subjects, subject 333 had 10,313 total positive reads, or positive control reads, and then subject 520 had 105,507 positive control reads. So this is the Kingdom classification results. This graph is a comparison of the 2 subjects at the Kingdom level, and as you can see, the types of organisms identified in the gut microbiome was 96.5% bacteria. So about 96.5% of what was found in the 2 gut microbiomes of the subjects was bacteria, and then less than 3.5% was made up of other, and other means that the reads of the bacteria were classified, but they make up less than 3.5% of our total reads and are not considered as important as our larger reads. Although we don't have any unclassified in this table, if we did, it would mean that the system recognized the read as belonging to the Kingdom bacteria but do not match any of the known kingdom. So these are the order classification results. And at this level, we start to see differences between the two gut microbiomes of the subjects. Subject 520 shows lower percentages of the bacteriodales and the clostridioides than subject 333. Subject 333 doesn't show any reads for the fusobacteriales and the Burkholderales. So yeah, the importance of this is that we start to see differences between the two gut microbiomes of the subjects. And then finally, at the genus classification level, we see significant differences between the two subjects. Here, subject 333 is about 60% of the bacterioids, while subject 520 is only 20%. And then differently at the Probotella, the subject 520 has a higher reading than subject 333, which is lower than 10%. And then there's readings where subject 333 shows less than 10%, and subject 520 shows no readings, and same for these three. So I think the importance of this is to see that there is a significant difference between the two subjects at this level. So our original question that we were researching for this experiment was, will there be a difference in gut microbiomes between a snake and a bearded dragon? And our hypothesis was that that there would be a difference between the two gut microbiomes of those species. However, since we were unsuccessful at extracting the DNA from the feces of the two different species, we were not able to examine their gut microbiomes, so we can neither confirm nor deny the validity of our hypothesis. However, if we are looking at the two different dog species that we compared from the database, they showed differences in the order and genus level. So we can say that they had a difference between their gut microbiomes.